Good afternoon, everyone. I'm Dr. Nico Rogelio, and I would like to welcome you to this month's PGH Grand Rounds, hosted by the Department of Ophthalmology and Visual Sciences. Today, we will be tackling an important and controversial topic, which is vaccine development for COVID-19. This webinar has been applied for zero point 0.5 CPD points, so make sure to sign the attendance and evaluation form via the QR code or URL posted to be eligible for the CPD points. Let us begin by singing our national anthem.
Mga kababayan, ang pambansang awit ng Pilipinas.
Kayang magiliw, telas ng silanganan, alam ng puso, sa titik mo'y buhay. Lupang hinirang, kuyak ka ng magiting, sa manlulupin, di ka pasisingil, sa nagatang putok sa simoy at sa langit ng bagaw, ay hinatang tula, tawid sa paglayang minamahal, ang isang ng matawad mo'y tagumpay na naghihingi, makituwid at araw niya ang kailan, pamay maghihingi, lumagang araw ng walhagit pagsinta, buhay langit sa piling mo, ang inigaya na pagkay mga amin, at mamatay ng dahil sa'yo.
Now, let us welcome the Director of the Philippine General Hospital, Dr. Gerardo Legaspi, for our opening remarks.
Good afternoon, everyone. I'd like to, first of all, congratulate the Department of Ophthalmology and Visual Sciences for this very forward-thinking topic for this afternoon, and quite, of course, very appropriate for the times. Though it's outside of their specialty, I think the mere fact that they chose it shows how how outside of the box this department really thinks, and I'd like to congratulate them for that. They have the DOVS has always been in the forefront of ophthalmologic advances and academic achievement for the hospital, and this afternoon's activity is another testament to that. The COVID-19 has been taking a lot of the good that we should be doing for other illnesses of our patients, but we have no choice but to still go through the motion of taking care of our patients and the challenges of COVID-19, but at the same time finding solutions on how to mitigate the circumstances to allow us to go back to ophthalmology or to neurosurgery and surgery, medicine, pedia, and all the other specialties that we have to, wherein we have to serve our patients, is a very important step to take. And I think the understanding of vaccines and the development of one is a must for all of us who are in the academy. So for this afternoon, I'd like to thank our speakers, Dr. Cruz and Dr. Gloriani, for giving us their time and their expertise in this much needed information that we have to have for fighting COVID-19. To everyone, my sincere gratitude for attending this afternoon's PGH Grand Rounds, and again to the Department of Ophthalmology and Visual Sciences, thank you very much.
Thank you, Dr. Legaspi. Before we begin our lectures, please be informed of our ground rules. All audience members will be put on mute during the entire duration of the session. Should you have any questions, you may ask them in the Q&A function below. Please refrain from asking your questions in the chat box. Please indicate to whom you are addressing the question to. During the open forum, we will try to get to as many questions as we can with the time left. Now joining me for today's Grand Rounds is Dr. Brian Messina, who will be introducing our first speaker. Brian?
Thank you, Nico. I'm honored to introduce to you today our first speaker, Dr. Philip Cruz. He was a graduate of the University of the Philippines College of Medicine. He had his residency training in pediatrics at the UP Philippine General Hospital, where he also served as chief resident. After specialization in 2001, Dr. Cruz went into clinical research, namely vaccine clinical trials, and also acted as a consultant in major pharmaceutical and biotechnology companies. Subsequently, he co-authored publications in peer-reviewed journals and presented in regional and international conferences and symposia. In 2010, Dr. Cruz took on a position with the GlaxoSmithKline Biologicals in Belgium as a senior manager in the Clinical and Research Division of Medical Affairs, Pediatric Vaccines. His scope was within the vaccine, medical, and scientific affairs of the emerging markets geographically from Africa, Middle East, Russia, South Asia to China. In this role, he had the opportunity to interact with ministries of health, technical advisory groups, and local experts to ascertain the public health burden of vaccine-preventable pediatric diseases. In 2013, he then took on a role as the area medical leader for Asia Pacific and North Asia, where he was responsible for providing a scientific case, including facilitating any necessary health economics, epidemiological, and clinical data to expedite the introduction in government immunization programs of new vaccines, which may, over the years, potentially save the lives of millions of children across the Asian region. Dr. Cruz was then subsequently assigned to his current role since 2015 to further support the clinical and medical affairs initiatives of GlaxoSmithKline in the United Kingdom and Ireland. Today, we are lucky to hear from the Medical Director of GlaxoSmithKline Vaccines for the United Kingdom and Ireland, Dr. Philip Cruz.
Thank you very much, Brian. Hope my sound is coming in clear. First of all, it's great to be home, though virtually. So hello, everyone. Good morning from London. Well, good afternoon right now in the Philippines. And it's a great pleasure to be here. Thank you very much, Dr. Legaspi. Thank you also to the Department of Ophthalmology and Visual Sciences for this great opportunity to share with you 10 months worth of clinical research of what has kind of plagued us now with the pandemic on COVID-19. So before I begin, let me just first of all share my screen and wish that my technology will be okay. Do let me know if my slides are coming through.
Yes, sir.
I hope you can see my slides. Okay, very well. Thank you. So the topic of my talk really is consolidating the amount of research, and the word unprecedented cannot be used enough of what's happening with us. And the overall theme is, with all these uncertainty, the only thing certain is science. And that's what we need to cultivate in our thinking, is to have that scientific methodology and that scientific approach. So before I go on to my slides, medyo hinukay ko yung aking baul, and just to share with you, 2 decades ago, I was doing my rounds along the halls of wards 9 and 11. This is a picture of my batch. It was taken obviously on post-duty for some, but yeah, so this is my batch in pediatrics. So I was a chief resident in the early 2000s. And next one shows you me in action. So this was taken in Ward 11, where I think this was a dry run for our Pedia 911 program at that time, which was kind of a groundbreaking, where you have all of those in a team-focused approach. And just to share with you, I remember a patient, gumaling naman siya, lumapit sa akin while I was doing rounds. And then, so these are my fond memories of PGH. So lumapit sa akin pasyente, sabi sa akin, Doc, yung pasyente nagsi-seizures, kaya binigyan ng phenobarbital. Sabi ko, ano? Yung pala, nagsi-seizure at binigyan ng phenobarbital. So, these are just the memories. So gumaling yung pasyente, don't worry. But yeah, really, I owe a lot to my training in the Philippine General Hospital. And this is the clinical acumen that I use every day as well in my line of work right now. So what I'm doing is what we call medical affairs, where we look at the science and try and translate it to vaccination programs. And my scope here is within the United Kingdom and Ireland. So it's a very challenging role. And what is basically an essence here is how we look at the science and the science of vaccines. So just a disclaimer here that the pandemic is a rapidly evolving situation, and thus up-to-date information may be available. However, I can guarantee you that information is up-to-date up until midnight last night. So it changes at a very rapid pace. And it's a quite complex topic at the same time. So I'll just focus on the general overview of the important aspects of this pandemic. So my focus is on three main points. First is immunology of COVID-19, vaccine development, and where we are with the current technologies, because it's quite daunting if you look at the current vaccines landscape. So let me just start off with knowing the enemy. This is the SARS-CoV-2 virus. So as you can see here, it has the conformation of many spikes. That's why they call it a coronavirus, because it's like a crown. And when you go inside, and a cross-sectional picture of this shows you the important components here. And when we design vaccines, we have to first identify what to target, because these targets are meant to produce antibodies, which are essential for protection. So I'd like to highlight the spike glycoprotein, which is the hotbed right now for vaccine targets. So it's those spikes coming out of the envelope of the virus. Next, you have the RNA and nucleocapsid protein, which you see inside the virus, and of course, an envelope. So these are the structures which we see would be pertinent and would produce an active immune response. And therefore, if you need a recipe for a vaccine, it has to contain any of these. But what we're looking on mainly is the spike glycoprotein. Some vaccines have the whole length of the spike protein. Some have a truncated or a shortened version of the spike protein. And just also looking at it in a 3D manner, think of the virus as a hand, where it has to have an open conformation in order to attach to a cell. And usually the attachment is on ACE2 receptor. So that's what we know. So the natural state of the virus is those receptors having a closed conformation. And then when it wants to attach to a cell, it opens. The reason why it's important, because that becomes our vaccine target. That close conformation which we want to maintain, and therefore have antibodies attack the closed, not the open conformation. So the secret to an effective vaccine is the vaccine antibodies that are produced will attack the closed or the pre-fusion state. So I've been asked quite a few times about the SARS-CoV-2 virus having mutations. Mutations occur. That is a natural phenomenon. And the research has a priority on the diversity and the evolution of the virus. However, what is comforting is that it has a low mutation rate. When you look at the RNA code of this virus, it contains 29,903 letters. And therefore, it is quite a lengthy code. So out of these, the low mutation rate happens around 2 letters every month. And in monitoring these mutations, what scientists have found out is that a significant mutation is on the 614th letter of the code, where D or aspartate changes to glycine. So we call this the D614 mutation. And what does this mean clinically? Well, it has been associated with an increased transmissibility. So mas madaling makahawa yung virus with the D614 mutation. However, we're still monitoring this in terms of, does it transmit more but have less clinical outcomes? So it seems that way. However, it's not a cause for concern for vaccine development. And also with regards to transmission, I'll just share the 3 C's. When you think of the 3 C's, that will basically sum up transmission. Crowds, enclosed spaces, having conversations, meaning, their distance is quite near. So in order to avoid transmission, think of the 3 Cs and MD, which is masks and disinfectants and distancing. So 3 Cs to avoid a trip to the MD. So these are important manners, because my message here is, when addressing this pandemic, a vaccine will not solve it. Not a vaccine alone will not solve it. It has to be a combination of these public health measures. Quickly now, what is in a vaccine? We have antigens, adjuvants, stabilizers, preservatives, and manufacturing byproducts. I'll be focusing on antigens, which is the part of the vaccine which creates the immune response. The others are likewise very important because, for example, the adjuvant, what it does is it enhances your immune response. Think of it like caffeine, a coffee in the morning where it perks you up. So it basically perks up the immune system to produce more antibodies. But what makes us unique? Why do immune responses to vaccines differ among individuals? So it's important to note that there are many factors. No two persons will have the same vaccine immune response. However, I just like to highlight here that age is important, and also the presence of comorbidities. If there are other medical conditions in the patient, that can affect the response to a vaccine. Age, obviously, as you grow older, in the elderly, the T-cell response is very much decreased, and therefore the immune system will not act as much when you were, let's say, 18 years old. So now let's go to vaccine development. The question is, when will we have a vaccine? Let me answer that with a number of slides. Looking at the history of vaccine development, the quickest on record for a vaccine development to occur was for mumps, where it took 3 years from discovery to when a vaccine became available. That was in the 40s. However, it's not the same across other diseases. Right now, we still don't have an HIV vaccine, though we're quite near. Others, coronavirus, no, not yet. HSV, herpes simplex, no vaccine yet. Malaria, it took 118 years for a vaccine from discovery. So it can vary. And it is really a complex journey. When you look at how vaccines are made, it takes anywhere from 10 to 15 years. 70% of production of a vaccines are dedicated to quality control. I'll give an example. The pneumococcal vaccine, before it goes out into distribution to a patient, it has to undergo 521 quality tests. Now, if it's at the 520th test, it fails, it will be thrown out, because you cannot compromise quality with a biological product. And therefore, the probability of success of vaccine developments is anywhere from 5 to 6%. So it's really not an interesting business proposition, I tell you. However, this is more on the public health proposition of preventable diseases and of the unmet need. And manufacture, it takes 2 years for some vaccines. Therefore, it really is a lengthy and complicated procedure. So the traditional model for current vaccines is broken down in terms of the timelines, as you see in this slide. So from the discovery research, the preclinical stage, clinical development, where you test it through phases, phase 1, phase 2, phase 3, where you look at safety, you look at efficacy, where you ramped up the trial into thousands of people to see other rare conditions if there might happen. So safety is of paramount concern here. And also of a regulatory review, because regulatory authorities need to look at your evidence and decide within these parameters whether they would approve your vaccine. However, this is not fast enough for a COVID-19 vaccine. Therefore, the proper term to use is to accelerate development. It's not about creating shortcuts, because that connotes a different mentality. It's acceleration. And before you can accelerate a vaccine development program, you need at least $2 billion for development, scientific research, and $1 billion for distribution. And therefore, what you see right now, when you look at the vaccine landscape, are partnerships. So, it's about a company merging with another company in their research, having a team-focused approach. So what we aim is the traditional timeline of 15 years, bring it down to 12 to 18 months. And how will that happen? So I highlighted here the areas where there could be these accelerated steps. So it's called development at speed. So what we do, we streamline the process, doing different stages of development and production at the same time, things happening in parallel. Actually, vaccines for COVID-19 are being manufactured as I speak. That's what we call manufacturing at risk. Because how can you produce hundreds of millions of doses after your clinical trials? The clinical trials are being done right now. It will end of this year, middle of next year. If you will only start making those vaccines, that vaccine will come out the year after. So right now, companies are actually making their vaccines. It's called manufacturing at risk. So it's not really an interesting business proposition again. So what are the possible efficacy outcomes of COVID-19 vaccine? Number one, it prevents death. Or the vaccine, number two, can prevent disease. However, it doesn't stop transmission. So nakakahawa pa yung vaccine. And third, an ideal vaccine prevents any illness, no fever, no coughing, anything, and the patient where the virus comes in will not transmit it. So that is a feature. Those are features actually of an ideal COVID-19 vaccine. So it can either prevent death, it can either prevent disease, or it can prevent transmission. So the third one is where you have the phenomenon of herd immunity, where you have most of the population being immunized and becoming immune. Now, the WHO gave a recipe for vaccine manufacturers at the start of the pandemic. They call it a target vaccine profile. So the minimal requirements for a vaccine has to be, it can be used in adults, including elderly, because they're very susceptible. The ideal efficacy estimate is 50%. So if the vaccine works in 50% of the time, okay na. That's WHO recommendation. And no more than 2 doses, because more doses makes it more difficult for vaccine implementation programs. And the protection should be at least for 6 months, because previous trials for coronavirus vaccines or even coronavirus disease, the antibodies are measured up until half a year to 1 year. Some rarity can last for 2 years. So the duration of protection is short-lived. However, when you have like, okay, a nice to have, now you have like the preferred is where you have a vaccine that can be given to all ages, at least 70% efficacy in single dose and protection for at least 1 year. Let me just focus a bit on efficacy. Let's say we have a vaccine that's 70% efficacious. And I'll give an example of what we have here in the UK, where 65 years old and above, the flu program has accomplished 75% uptake rate. So 75% of the 65 years old and above here in the UK is vaccinated against flu. So the uptake is 75%. If you have a vaccine that is 70% efficacious, and you give that to the 65 and above, so 70 times 75%, you only have 53% efficacy. So it means that half of that population will still be vulnerable with a 70% vaccine efficacy. So you can see that the point that I'm making, that a vaccine is not the silver bullet. It has to have a team approach of public health measures, mass social distancing, and other guidelines that needs to be in place. In vaccine development, what are we watching out for? Safety is a very much a priority. There's no compromise on safety. And what we're watching out for is what we call antibody-dependent enhancement. So what happens is when you produce antibodies against a virus or bacteria, there are 2 types of antibodies that are being produced: neutralizing and non-neutralizing. Neutralizing basically takes care of that virus. It eliminates it completely. A non-neutralizing antibody is what we also call a binding antibody. So what it does, it binds to the virus, okay, and acts as a bridge and links up your immune cells. What it does, it bridges the immune cell to the virus, and then the virus goes to the immune cell, produces more virus. So instead of producing benefit, it produces harm. So that has happened with other vaccines in the past. In 1967, an RSV, respiratory syncytial virus vaccine, was given to infants, and it created this phenomenon, and 2 babies died, unfortunately, from this exaggerated immune response. Instead of causing neutralizing antibodies, it produced the binding or non-neutralizing antibodies. And Thankfully, so far, there's no evidence that shows that SARS-CoV-2 can have this phenomenon. And by the way, another virus that exhibits this is dengue virus. So let's just now go down to technology. This one, I needed to check every hour last night to make sure that I have the latest data, because every day changes. There are a total of 198 vaccine candidates for COVID-19 globally. However, 44 of those candidates are in human clinical trials. The rest are in the preclinical state or still looking at early phases or early research. So quite daunting, very much unprecedented. I'll just focus on the technologies in a broader sense, because when you look at the menu of the vaccine candidates, the technologies are really, sounds like science fiction, but really this is the future. And COVID-19 is the catalyst for us to look at these technologies, which otherwise we were looking at future technology, but now we're bringing it forward. So there are four main categories of vaccine technologies for COVID-19. The first is a whole virus, which is a traditional technology. You get the virus, take away the infectious causing property, and then put it in a vaccine, and then you produce antibodies. However, the limitation here is you have to have a bit of an adjuvant sometimes to increase the immune response. Same with protein-based. So what you do, you take the SARS-CoV-2 virus proteins from the spike protein that I showed you earlier, purify it, and then put it in a vaccine, and put in also an adjuvant, and you give it to a patient. So what it does is basically put in the fingerprint or the recipe of the virus, so that when it is injected into a patient, the antibodies are produced from those instructions. Next is a viral vector. Viral vector is quite an interesting technology, because what it does, it uses viruses as transport mechanisms. So there are certain viruses which are great to be vectors. So it's like a shell. So it's a vehicle, and then you put in the code of the RNA virus, which is SARS-CoV-2, into this vector. So basically, it's splicing and dicing. Cut the code, put it into a virus. Usually, what we use is the RNA adenovirus. Put it into the adenovirus and inject it to a patient. So these viral vectors are the leading candidates of what we see right now. So a limiting factor here is previous immunity to these viruses. So therefore, what is a consequence is you don't produce as much effective antibodies as you would. However, there are certain components there that could somehow boost those immune responses. And the last one, which is cutting edge, basically, is the nucleic acid technology. So what it does here, it looks into the RNA code or DNA, RNA code of SARS-CoV-2, take out the code for the spike protein and put it into a vehicle as well. So what it does is you create software and then inject it to a patient, so that the vaccine, the software goes inside the cell and creates antibodies from within your cell. So the first two technologies produces the vaccine in a factory or a manufacturing site. The last two produces the vaccine inside your body. So think of it as 3D printing a vaccine inside your body. However, the caveat here is this technology hasn't been proven yet, because there's no vaccine that has DNA and mRNA nucleic acid platforms as of yet. So this is very new, and therefore we're watching with bated breath on the clinical development of these and the data. Data looks promising, because what we are monitoring are safety concerns, and it's more or less tolerable in terms of their profile, and we're now just monitoring and looking at the efficacy. So the data have yet to be published, and so what I would suggest is to wait for peer-reviewed journals, because right now, since it's unprecedented, you have publications which are not yet peer-reviewed, but broadcasting their data. However, like I said, the proof is in the data. There are some vaccines which come out with press releases before their clinical research data. So I would urge you to exercise caution when looking at these vaccines. So what's the global strategy? Will the Philippines get a vaccine? Will the African continent get a vaccine? Will obviously the other countries will do. But the global strategy is all countries vaccinated with some of their population is much better than some countries with all of their population. Because right now it's a pandemic. If a country vaccinate all of their population without minding others, with the world right now where travel is basically everywhere, it's not going to be a worthwhile strategy. Therefore, you have institutions and groups, supranational groups, that will take care of vaccine allocation throughout the world, because it's more or less having broader coverage, not just in a certain country, but everywhere. However, discovering a safe and effective vaccine is just solving half the problem. Why is this? Cold chain. How do you distribute millions and millions of vaccines? And with the mRNA vaccines, it has to be minus 80 degrees Celsius if you were to store it beyond 3 months. So it's very, very complicated in that sense. And it's important to highlight how important it is to maintain the cold chain. Because what happens if you have a cold chain break? Might be your vaccine becomes ineffective. And then you give that vaccine to a patient gets COVID, and people blame the vaccine, where in fact it was not stored properly. So these are important things to consider. So I'll end with this slide that shows you the unknowns. So I showed you what we know, and now I'm presenting to you what we don't know. What is the level of neutralizing antibodies to confer protection? We don't know. So what is happening right now is we're looking at convalescent sera, those patients who got well from COVID-19, measure their antibodies and do assays and basically relate it to the vaccine data that we are now generating. However, we don't know yet the level. Will all vaccines in development make it through the finish line? No. History tells us otherwise. So it's like for vaccines, it's 5 to 6% get it through the finish line. However, everyone is rooting for all the vaccine candidates to make it through the finish line. What is the contribution of other immunological responses in protection? So that's one thing that's very interesting, because you have studies that look at MMR vaccine, BCG vaccine being given to patients for COVID. So what basically the theory is more the non-specific immune benefits of having these vaccines being given to patients. So it stimulates the immune system broadly. So that's what the BCG and MMR vaccine kinds of aims to do. However, it doesn't mean that you had BCG before you'll be protected. No, the studies that show right now is like a 1 year kind of duration from when you receive the BCG. That's when they theorize that your immune system will act in the sense to at least have some form of protection. Antibody-dependent enhancement, the one I highlighted earlier, we don't know. Thankfully, we don't see that phenomenon as of yet, and hope we won't see it. So that's more of the safety monitoring. That's why vaccines need to be monitored on their safety. Last night, the FDA issued guidance that once a vaccine gets rolled out, safety surveillance should be at least 2 months after when the vaccine is administered. How will the pandemic evolve? Will SARS-CoV-2 and thus vaccination become seasonal? They say that and I know the news that when summer comes, or when the temperature rises, there's going to be less COVID-19. However, the research, and these are generated research on temperature and humidity, which shows that the higher humidity and the higher temperature does not favor the virus. So it's theorized to decrease in that settings. So those are the unknowns. And I guess really what the answer to the questions is, when will we have a COVID-19 vaccine? And before I go to this statement, which I'll quote from my research, we're looking at programs, and you might hear of emergency use for COVID-19 vaccines probably within this year, end of this year, our side of Christmas. we might see a vaccine being implemented in the US, in the UK as well. However, for the UK, I'll give you an example. We're probably looking at a vaccine implementation in Q1 or Q2, early Q2 of 2021. So to answer the question, we will have a safe and effective COVID-19 vaccine when the research studies, engagement processes with authorities and regulatory authorities and public health authorities and departments of health, communication and education efforts undertaken during the clinical trial stage have built trust and result in vaccination recommendations being understood, supported, and accepted by the vast majority of the public, priority and non-priority groups alike. So it has to be a team approach, a community approach. And therefore, I'll end the slide with an appeal to have a bayanihan approach, because that, I think, would be the answer to the pandemic. It's not just the vaccine, because there's no one vaccine that will address this. In fact, what we in industry, in the pharma industry, want is a lot of vaccines. We don't want all eggs in one basket. We want the vaccine platforms to be as varied as possible, because one vaccine has weaknesses, the other has strengths. So it could be a scenario that you have two vaccines being given to a patient, two different vaccines, because it utilizes the advantage of each. So that's what I can say of the future from the present. Thank you so much. And yeah, I'll hand it over to the facilitator. Thank you very much.
Thank you, Dr. Philip Cruz, for that very informative lecture. Once again, I'd like to remind our audience that we will be addressing questions only during the open forum. Until then, you may ask your questions at the Q&A function below. I now turn you over again to Dr. Brian Messina to introduce our second speaker.
Thank you again, Nico. Our second speaker obtained her BS Public Health degree from the Institute of Public Health and her medical degree from the UP College of Medicine. She obtained her PhD in microbial immunology from the University of Melbourne in Australia. After this, she received the Fogarty Fellowship Grant for postgraduate training in HIV and AIDS, immunology, and virology from the University of California, Los Angeles. She also attended various trainings focusing on biotechnology, hepatitis B virus, sexually transmitted diseases, and clinical microbiology from different parts of the world. She came back to our institution to serve as a professor at the UP College of Public Health, and she became chairman of the Department of Medical Microbiology, College of Public Health, and director of the Institute of Biotechnology and Molecular Biology of the National Institutes of Health in 2005. She served as the Dean of the UP College of Public Health from 2007 to 2013, and at the same time period became the Center Director of the CINEO CHOPMED Regional Center for Public Health, Hospital Administration, Environmental, and Occupational Health. Dr. Gloriani has received numerous awards, most notable of which are the UP Alumni Association 2006 Outstanding Professional Award in Public Health and the PSM Outstanding Microbiologist Award in the same year. In 2019, she was declared as the regional and national winner of the PCHRD Best Mentor Award. In this fight against COVID-19, Dr. Gloriani holds a vital role as chairperson of the Vaccine Development Expert Panel of the Department of Science and Technology. Ladies and gentlemen, let us all welcome Dr. Nina Gloriani.
Good afternoon, Director Gap and everyone, and thank you to the Department of Ophthalmology, especially Dr. Corina, for inviting me here. And I'm very pleased to be with the medical doctors and actually the young ones. Compared to Dr. Philip, I'm way older. So may I share my screen now? Okay, I will share this one. Okay. Thank you very much, Dr. Philip, for that wonderful presentation. It has set the tone for my presentation. So I've been given this topic, strategies for COVID-19 vaccines, clinical trials in the Philippines. So just this outline, I may just give the landscape. Dr. Philip already updated his slide. Mine is last October 15, but anyway, we're going to look in one slide about older technologies versus novel vaccines. And then I'll move into the clinical trials, phases 1, 2, 3, and the clinical trials in the Philippines, and specifically an overview of the World Health Organization Solidarity Vaccine Trial from the global and national perspective. So I'll give you some idea of what's actually going on, what we are preparing for this COVID-19 clinical trials here in the Philippines, and end with my take-home messages. Okay, so October 15. This is the landscape of the WHO. Please note, Dr. Philip said 44 already by October 19. Here it's only 42. So every day it changes. Something is added every time. But take note that most of the vaccine trials in the clinical phases 1, 2, or 3 are using the protein subunit platform. But we will take note later what are actually currently available and which ones are in the more advanced phases of development. So just looking at this, actually, this is the New York Times. It's somehow different. The statistics they have is a little different despite the same date. So I think Dr. Philip already told you about the many stages of the vaccine development. And here I have summarized the COVID-19 vaccine platforms. Also, Dr. Philip beautifully explained that you could have the whole inactivated vaccine, the virus. You can actually weaken it, or you can just use parts of the virus. And most of the current vaccine candidates actually are focusing on the spike protein because that is the one that will attack the ACE receptor. So this is all again as of October 15. I'd like you to consider that there are 10 in phase 3 clinical trials. And here in the Philippines, we're actually monitoring all of these 10 and maybe the newer ones that will come in. So for the messenger RNA vaccines, we're monitoring closely Moderna and Pfizer. These are from the United States. The DNA vaccine from Novavax. And vectored vaccines. These are use adenovirus either 26 or adenovirus 5. So we have at the top is AstraZeneca. Maybe you hear about Sputnik V here in the Philippines. We're also evaluating the Gamaleya vaccine. The Janssen vaccine is also a vectored vaccine, but we're not actually looking at the CanSino Biologics vector vaccine. Then we have 3 inactivated vaccines from China with whom we have a confidentiality date of data agreement with the Philippines. So these are the 10 ones. But we're not close to others. We're actually considering many other vaccine candidates, and they have actually are starting to have to forge bilateral agreements with the Philippines. So the preclinical development, as you all know, will require, well, of course, laboratory studies and then animal studies, proof of concept, safety studies, potency assay. And before you can move to phase 1 to the humans, all of this in the preclinical phase should be really successful. You know, all the parameters should be right, and then you move into phase 1 and phase 2. So in the Philippines, only vaccines that have completed the preclinical and of course the clinical phases 1 and 2 trials will be evaluated and approved for phase 3 trials in here. So please take note, only phase 3. We will not allow phase 1 or phase 2. Maybe phase 2 moving into 3, maybe. But generally, we're talking about phase 3. So what do we test when we consider these different phases of the human clinical trial? Well, first and foremost, of course, we know it's safety. We're looking at local and systemic side effects. For the most part, there will be mild such adverse side effects. There may be moderate adverse effects, and already you see on the news, you saw on the news about AstraZeneca's having paused its clinical trial phase 3 and recently Janssen. So, but of course, these are being investigated right now, and we are monitoring closely what will happen and what will be the final findings. So aside from safety, that is usually during phase 1, that tests only a few individuals, maybe 10 to 50 to 100. Then it moves to phase 2. That will also include safety as well, but more on dosaging, immunogenicity, and when to give the vaccine. So we're looking at whether the vaccine is also able to generate immune responses, largely neutralizing antibodies, and also cell-mediated immune responses, largely T-cell responses. So this is now done in about 100 to 1,000 volunteers. So looking at optimal dosages. So if you have the optimal dosage and when to give, that's when you move to phase 3. Phase 3 is efficacy. You want to find out if the vaccine will protect against COVID-19 disease. And Dr. Philip has actually presented the different parameters for efficacy. It could be against death from COVID, against mild disease, or even just against virologically confirmed COVID. Okay, so phase 3 will also involve safety and immunogenicity as secondary endpoints, and will now involve thousands of volunteers of diverse ages, race, and special populations. But after the phase 3 and everything is okay, then the vaccine is registered or licensed. I'll show you later what happens here in the Philippines. But still, after administration to a large population, the vaccines will still have to go through phase 4. This is called post-marketing surveillance for rare adverse events as vaccines are rolled out in the general population and special groups. Okay, so phase 3 for clinical trials here in the Philippines. So what are we looking at for this COVID-19 phase 3 clinical trials here in the Philippines? There are two types of vaccine trials. The first one is the WHO Solidarity Vaccine Trial, which is supported by WHO and largely by the Department of Science and Technology and the Department of Health. And the Philippines has officially submitted its intent to participate in this big trial and has nominated the principal investigator to oversee the Solidarity Trial. So Dr. Jodor Lim of the Infectious Disease Section of PGH has been named the overall principal investigator of the Solidarity Vaccine Trial. But aside from the Solidarity Trial, there are also independent clinical trials. And this will fall into two types, the ones where we have bilateral partners. I'm sure you are very familiar with our partnership or agreements with Russia and with China, but we also have other agreements with Australia, Japan, USA, and I think Canada later also. Then some private vaccine companies can actually also conduct phase 3 clinical trials here in the Philippines. Now, for both, there will be the same rigor in approval process, but I'll go into that in a little while. Okay, so what is the Solidarity Vaccine Trial? This is an international, multi-center, so all over the world, double-blind, meaning the people who receive the vaccine do not know what they are receiving, whether it's the vaccine 1, 2, 3, 4, or the placebo, which we call a comparator vaccine. And the one administering the vaccine, the doctor, the nurse, will not know what that vaccine is. So it is so we say double-blind, placebo-controlled, randomized trial of candidate vaccines against COVID-19. And so far, more than 200 trial sites around the world have expressed interest in participating, including the Philippines. So the World Health Organization has 6 geographic regions. So Africa, Americas, Eastern Mediterranean, Europe, Sharo, and WIPRO. We are in WIPRO. So the Philippines is classified under WIPRO. So we should be in the map of the Solidarity Vaccine Trials. So what is this all about? This is an international randomized trial of several candidate vaccines at the same time. So 4 things here to consider. So the Solidarity Trial will evaluate several different candidate vaccines, which we hopefully will increase the likelihood of finding several effective vaccines within a short period of time. Well, actually, But so far, I have to update this of today, is 23rd, no? The vaccine candidates have not yet been identified by the WHO, so we have some delay. And then another aspect of the Solidarity Trial is that it will allow expeditiously enrolling participants at sites with high rates of COVID-19 and resulting in rapid accumulation of data to support a rigorous evaluation. It will also eliminate the inefficiency of designing and conducting separate trials. And as I said already, the results could be available within 3 to 6 months after each vaccine is ready for infusion. And last but certainly not the least, there will be that sense of international collaboration and countries' commitment. And it will foster international deployment, hopefully with equity of access, because availability does not mean access. So we hope we will have access to the vaccines once they become available. So what is this Solidarity Trial here in the Philippines? The WHO Solidarity Trial is an adaptive trial and will focus on high transmission areas to vaccinate people. No? So we will call this the trial zones. So there will be fixed trial sites in hospitals or in community health stations that we will identify. For instance, in the National Capital Region, we are now working closely with communities, with the mayors, with the LGUs. But an additional special feature of the Solidarity Trial is the possibility of setting up mobile trial sites, or what we call pop-up sites. Now, the pop-up sites are based on the concept of ring vaccination. Ring vaccination is an infection control measure that involves vaccinating a cluster of individuals at high risk for infection based on their social or geographic connection to a known case. So if the case is here, the middle here, everyone around, the closer, so you have this first layer around the case, a second layer, and a third layer. So this is the concept of ring vaccination, which was actually followed or used in the testing the Ebola vaccine during the Ebola outbreak. So this ring vaccination will create a protective ring or cluster of immune individuals around newly diagnosed cases. So I will explain that again later when we discuss which barangays could be potential trial sites for the Solidarity Trial. Now, because this is a multi-center, or a global trial, we are only expected to to enroll maybe around 2,000 participants, although WHO asked us to double that to 4,000 for the entire Philippines. And the priority group to recruit is those 18 to 59 years old, high risk for COVID-19 infections, such as the healthcare workers, frontline workers, and as I have said, the contacts of cases. But special groups may be included later, such as the elderly, 60 years and above, children, as well as the immunocompromised. So there will be a separate protocol for these special groups. And as you all know, when you do clinical trials, there will be inclusion and exclusion criteria defined. When maybe if you just look at exclusion criteria, those who have had COVID cannot participate anymore. That's only one of the exclusions. There are many other exclusion criteria. And the randomization of vaccines will be done by the World Health Organization. Right now, we do not know how many vaccines will be tested. It could be from the last meeting we had, around 4 have been, well, actually 8 have been shortlisted, but 4 met most of the WHO target product profiles, plus a comparator placebo. And the immunization schedule could be 1 to 2 doses, well, preferably 1 dose, but most of the vaccine platforms that I showed earlier use 2 doses. I think only AstraZeneca will use 1 dose. All the rest use 2 doses of intramuscular injection within a 14 to 28-day interval, depending on the vaccine type. So the vaccine expert panel under the DOST is actually looking at each of these, evaluating each of these vaccine types for their compliance with the requirements that we have set. As we know, vaccinated individuals will be monitored regularly. And just in a nutshell, the primary outcomes, the efficacy, because clinical phase 3 is efficacy. The primary endpoints uh, will come within 3 to 6 months after immunization under the Solidarity trial. But the secondary outcomes, all the like safety and other immunogenicity parameters, all participants will be followed up for up to 12 to 18 months. Okay, so how did we, or which sites were actually chosen to join the Solidarity trial? So we have set minimum requirements for clinical trial sites as well as investigators. So the initial participating sites were expected to come from high COVID-19 transmission areas, such as the NCR. Well, at the time that we were doing this, Metro Cebu had a lot of cases. Now Metro Davao and I think Western Region 6 has a lot of cases also in Bacolod, and some pockets in Region 4A and Region 3. Now we need updated CVs of principal and co-investigators and the rest of the team, their current good clinical practice certificates. Now the updated PRC licenses are not required by SJREB or the ethics boards, but the VEP required this following the US FDA requirements. They should be willing to sign FDA confidentiality data agreement and should have no conflict of interest, no attachments or affiliation with any of these vaccine companies, for instance. They should be affiliated with accredited hospitals and laboratories with accredited facilities for COVID-19 diagnostics, especially for RT-PCR testing. Now, why? Because this is an efficacy trial and all participants, the vaccine participants who develop signs and symptoms of COVID will have to be tested for RT-PCR, for the virus. So they have to be in the facility where this is available. So that is a requirement, and all such hospitals submitted their accreditation for COVID-19 as COVID-19 diagnostic facility. Also, they have to be accredited by FREB at level 3. So, but hospitals which are not level 3 accredited may affiliate with level 3 accredited hospitals if they also want to to join the trial. Now, they should be able to integrate clinical research space into their existing clinical practices, provide space for vaccine storage, archive space and equipment, as well as workspace for clinical research associates and clinical research coordinators. So that is why we need the heads of the hospitals to actually approve this participation, because we need their their approval to consider giving this space or allowing them to store the vaccines in the pharmacy and so on. So we are looking at all of that. And this is just refers to the CROs who also need to submit their license to operate, their company profiles and other DOH, FDA, and WHO requirements. We're just looking at the target product profiles for emergency vaccines clinical trial at this time. We're not looking at the really routine vaccines. Now, just to give you an idea of these hospital sites that have been identified with COVID-19 testing laboratory accreditation and level 3 research ethics board accreditation, we put them into clusters. So cluster A is PGH and Manila Doctors Hospital in Manila. They each will recruit anywhere from 150 to 200, maybe up to 400 if we can produce enough funds for the additional 2,000 that WHO requested. So another cluster is B, also in Manila, San Lazaro. Lung Center and St. Luke's Medical Center QC will be in cluster C. RITM and Makati Medical Center are in cluster D, and Medical City and St. Luke's Medical Center, PGC, are in cluster E, and then the others are in the other regions. But so far, what we are, we're preparing the sites for the NCR more than the others. Maybe the others will be joining in the later phases of the solidarity trial, because what will happen is a few vaccines will be tested first. Let us say 2, 3, 4 vaccines, and then if some of these vaccines are not working or found ineffective very early on, new ones may be tested, may be added to the vaccines that will be tested. We're also currently considering inclusion of Region 6, Iloilo, Western Visayas Medical Center. I think Bacolod is in Region 7. We now have ongoing talks with the Resuhead and the hospital, as well as the Medical City Satellite Clinical Trial Unit in Iloilo. Okay, so this gives you a colorful, at least, map of the sites chosen for the Philippines. We were the ones who chose this. It's not yet approved, but this is the one that we will present to, we have presented actually to WHO. So it's color coded. So for Manila, actually we have the barangays have been identified. Also for Quezon City for each of these. But for Cebu, for Cavite and Davao, we don't have the trial sites yet. And what this shows here, that why we had to map like this and cluster them, because they will be working very closely with defined communities at the barangay level. Okay. So we're working very closely with the Department of Health Epidemiology Bureau in tracking the epidemic. So we're looking at cumulative cases, active cases, and attack rates that they provide. And we put this into maps so we can identify the darker, of course, the color, the more cases you have there. The thing is, when we started doing this, that was way back in September 1, there were more of these dark colored spots here. But now, although we keep getting 2,000, more than 2,000, 3,000 cases per day, most of the areas, or some, not really most, some of the areas in the NCR, for instance, have no new cases anymore. So but other cases, other areas have more cases. So we're tracking this now. So we have weekly or every 2 weeks data from EB that we should be doing this. So the trial sites or zones will be the top 5 to 10 barangays with high COVID-19 case rates that are close to where the hospitals are. So the catchment area, we call this the catchment areas or catchment barangays of each hospital. So just to give an overview of the trial process, this was already discussed with vaccine teams, with the global trial team. So this is very much technology. We will use a lot of technology. So in these COVID times, you cannot have so many people being oriented on the trial. So we have to really strategize and plan very well on how we will do the group information on ICF. Currently, what they're thinking of is only 10 people in the morning and 10 potential participants in the afternoon, and following all these social distancing precautions. Then they will go through the different about these processes here: trial ID, pre-screening, informed consent, inclusion, exclusion criteria, biometrics, and so on. So everything will be electronically generated. They will be using the iris technology to identify participants and barcoding everything. I think we're familiar with this, but all of the vaccine teams will be provided with, well, of course, the software, but and also the tablets, all the gadgets that they could possibly need, that they would need. So in addition to all of those, there are what we call global participatory practices that the World Health Organization provides us with, the framework for this GPP. And before the trial starts, they have all of these things that we have to consider, that we should be developing a succinct and evolving stakeholder engagement plan, an operational communication and issues management plan, and stakeholder engagement plan in all aspects of the trial preparation. And we are doing this with the Philippine Department of Health, Health Promotions Bureau Communications team. So the DOH HPB has uploaded most of the materials they have prepared. Also, I'll show it to you later. And that will be updated regularly as we go along. So we are in the pre-launch phase. And for the last part here, the fourth aspect here, to set up optimal trial conduct, the study research team, coordination and communication structures, and logistics system, the vaccine teams, all 10 of them under Dr. Jodor are working very hard to put in place, to comply with all the requirements that the FDA wants. No? But again, also very early on, liaising with the local government units because that's where we will get our participants. Okay, so I want to show this. Are we ready? Actually, we're trying to be ready. We have been preparing hard since, I don't know if you know, but the vaccine expert panel was convened way back in March 2020. We started evaluating some of these vaccine candidates at that time, very early at that time, and then help set up or streamline the processes for the um, evaluation. But for the WHO Solidarity Trial, they have the site's readiness milestones that we need to follow. So at that time, the local site PI is designated. Even before August, we already designated Dr. Jodor to be the overall PI. But maybe what will be important here is that all these three with the red arrows, we're doing all of this. We're coming up with the epidemiological information, local disease incidence and deaths, zero prevalence studies. Although we lack data on zero prevalence studies, we hope we can get some of the doctors or those groups who have information on that to provide us so we can submit to the WHO. The antibody positivity data will be required. And then, as I have said, the site-specific, the vaccine teams in their specific sites are looking at the SOPs and what the specific IRBs will require from them. Although we do have generic protocols and generic documents to submit. And the laboratory aspects are also being ready right now. So like, we hope we will not get the minus the messenger RNA vaccines, the RNA vaccines, because they will require minus 80. But in case we do, we are also getting ready for that. So we have the sub-TWG on vaccines under the Department of Science and Technology. And it does not just look at the technical evaluation, it looks also at the procurement and all the logistics involved in this, at least for the Solidarity trial. So the pilot run of the Solidarity trial was supposed to start third week of October. That was the initial schedule, but it was delayed to 3 to 4 weeks. And just, when was that? I think Tuesday, we learned that it will be delayed for another 4 weeks. And that we hope to start in the Philippines, the trial in December. So we might have some problems there. Okay, I just wanted to show also this, there are 6 panels here, to show you what the DOH HPB, Health Promotions Bureau, is doing. They have already this stakeholder engagement plans, including communication plans for the pre-launch, the launch, and the post-launch. Just to say that there has to be one voice. What are we trying to say? That in government, the top heads should be saying one thing and not another. Like if we, all others, academic partners, professional societies should be one, giving the same message or a similar message or one key message and not contradicting each other. So all of these have been laid out. There are key communication approaches and strategies. So you have your partners and influencers, health workers, general public, policymakers at the national and local levels, especially at the local level when they start announcing that, let us say, 10,000 participants from this province will be involved. That is not true. We think that these local heads should not be announcing those things. Okay? And then we are looking, of course, at media practitioners. So there are many questions from media, and we need to somehow manage those questions well so that we do not give contradicting answers. So we have these stakeholder audiences. Office of the President is included there. The IATF, the National Task Force, local and chief executives. We already presented to the NCR mayors the plans for the Solidarity Trial, and they were all very supportive. And in fact, um, volunteered some of their sites. Okay, so we have the sophisticated lay audience, general public, the scientific community. We're looking at people who could help us communicate. We're looking at the professional medical societies who will be our partners here. And then there will be this implementation plan. And so far, the timeline of activities, they're on track. Ang nadi-delay ay yung WHO. So yun pong ating preparations, medyo okay tayo. Now, just a few more slides before I end. Now, for everyone to know, especially you doctors, no, that there are approvals at various levels. We already said that whether you're doing the Solidarity Trial or you are doing it with independent vaccine companies or the private pharmaceuticals, you have to go through certain requirements. So already I showed you what were required of the hospital sites and the investigators. So all of these complied. Then the clinical trial application will also pass through the ethics review boards. Now, under the Solidarity Trial, we're looking at the single joint research ethics board, but all other site-specific IRBs are now required to join the SJREB. So that will facilitate the approval process or the evaluation process for everyone. Then we follow WHO target product profiles as already mentioned earlier in the SOPs. And finally, the Philippine Food and Drug Authority will have the final say. It will be the one saying it will be registered or licensed. And once, well, actually not the licensing of the vaccine yet for commercialization, but for the approval of the clinical trial to proceed. So tentative timelines, I have changed the October to December 2020 to June 2021, looking at the 3 to 6 month primary endpoint timeline, but this may be extended. Actually, the DOST will support the Solidarity Trial for 18 months. So we're looking at up to June 2022. So what are these things that we need you to submit if you want to be involved? So the WHO has all this list of documents, very specific SOPs. So right now we do not have the final documents from WHO, but we're trying to produce the harmonized one among the 10 sites, and including the informed consent form which the ethics boards will require. So the application process has been streamlined. It went through several, not just days, but weeks and months of changing and streamlining the application process, because the COVID-19 is a special problem. The COVID-19 vaccine is a special vaccine. Instead of going into the FDA directly, the DOST created us, the vaccine expert panel, to do the preliminary screening on these vaccine candidates. So you note here, first, the applications for clinical trials are on a per protocol basis, meaning a company could apply under the Solidarity Trial or independently, each one, Solidarity or independent, with its own study protocol, treated as two separate applications. And the list of documentary requirements is listed in the DOH AO number 2020-0010 approved last March 2020. That is available online from the DOH website. So the additional step before FDA, normally under normal circumstances, you go directly to FDA. The additional steps are here, and that's about 14 days. So there is a VEP submissions group at PCHRD that receives applications and then looks at for completeness. If the documents required are complete, then the documents will be submitted to DOST VEP for the technical evaluation and in parallel to the single joint reb so that the process of evaluation, this is for ethical, this is for technical, will be faster. So we're given 14 days to do that. So what we do actually, we had the test case before. So we were able to finish an evaluation for one vaccine candidate in 7 days. And then SJREB also in 1 week's time. And then we have to come together to maybe resolve differences if there are any, or agree on what trial zones will be assigned to such application before we send to the FDA. So the FDA will have longer days. There are more documents to submit to FDA. And another regulatory review. FDA has its own scientific advisory committee. So, but this is preliminary that we do. Actually, what we say may actually be already considered by FDA and could fast track or facilitate, expedite further the process. So, but in addition to all of this, the DOH Centers for Health Development also has reportorial requirements which can be found on the online on their website. So for this approval process for clinical phase 2 trial, whatever the platform technology used to generate the COVID-19 vaccines, whether old or new generation technologies, the inactivated vaccines of China or the adenovirus vaccines of Sputnik V or AstraZeneca or Moderna, the investigational vaccine product has to satisfy standard criteria and pass stringent tests, from vaccine discovery to preclinical tests in vitro and in animals, and finally in human trials. So the final stamp of approval is the FDA's approval. So my last slide, my take-home messages. There are really multiple approaches towards the same goal, that of producing a safe, effective vaccine against COVID-19. Actually, the question now, even among our politicians, our heads in the different line agencies or in the LGUs, are we going to choose the vaccines from older technologies, or would we want to consider the newer platforms like messenger RNA? So we're studying the advantages and disadvantages, the pros and cons to each of these types. But what we're looking at really is probably a combination of both old technology and next-gen platforms. It is very likely that different vaccines, not just one type, will be needed for the pandemic response. And multiple companies with different vaccine candidates now are working together to meet that global demand. After all, not one single vaccine developer can produce the amount or the volume of vaccines that we will need. There, we have to highlight the need for scientific rigor and integrity in approving vaccines for licensing and commercialization. And as a public health person, I really would like to see all of these vaccines having an impact to public health. So we need monitoring after administration. So we do, of course, that's required, post-marketing surveillance for safety. But at the end of the day, at the end of 1, 2, 3 years, 5 years, we should be looking at whether what we have been doing or our choices will have any impact to public health. So with that, I end the presentation and thank you very much for your attention. Okay, stop.
Thank you very much for that wonderful lecture, Dr. Gloriani. We will now get to our audience questions in our open forum. While the open forum is ongoing, you may still ask your questions through our Q&A function below, and we'll try to get to them. Right, so here we have some questions. Our first question is for Dr. Cruz. I read that inactivated vaccines can lead to disease enhancement effect, where vaccine-triggered antibodies, instead of offering protection, could worsen infections when people become exposed to a virus after inoculation. Please comment.
Yeah, thank you very much for that question. And this is a topic that I discussed in my talk. However, just to highlight that disease enhancement, we see it in viruses. So a prime example is dengue viruses. There's the feline coronavirus and RSV, or the respiratory syncytial virus. So that is basically what we're watching out for in all our clinical trials. And we don't see it at the moment, thankfully. However, it's still a possibility that, this is a theoretical possibility. So the important thing is we know what to watch out for. Is there any severity of disease? That's why during the conduct of a clinical trial, you have safety monitoring in place. You have a committee of experts, clinical experts, looking at any signal that might signify that there is an enhancement going along. So far, none. I can go through, Nico, if you want, para mas mabilis. Marami tayong makuhang questions. So next question is, can you comment on herd immunity? If I'm not mistaken, the Spanish flu was controlled with herd immunity along with the implementation of minimum health standards. So for the audience, the Spanish flu was the pandemic in 1918, where 50 million died. So that's the thing. If you don't have a vaccine, that's the possibility. So during the Spanish flu, there was an exposure of 1/3 of the world's population, and 50 million died. Right now, there's less than 1% of infected for the whole population. So we don't want to go with that statistic. And therefore, we need to adopt all of these measures, public health measures, and eventually a vaccine. We don't want herd immunity. It's not a strategy, it's a phenomenon. So herd immunity is not a strategy, just to be clear about that. And so nangyari sa Spanish flu, the virus ran out of susceptible hosts. So it basically died down. But then that's a different topic altogether, because those who survived actually during the Spanish flu were an interesting fact is most survivors were in the 30-year-old range, because apparently they were exposed to previous flu epidemic. It's called the Russian flu. I think it's a pandemic at that time. And so they were given some form of immunity. So that's one phenomenon also I'd like to share, that there is cross protection amongst viruses, which can help your immune system combat this disease.
Thank you, Doctor. Yes, sir.
Yeah, go ahead, Nico.
Dr. Gloriani, would you like to add anything about herd immunity?
Yes, actually, there's some controversy there. Some people would prefer to have herd immunity from natural infection. For COVID, I don't think that's a good idea. What we in immunology, what we're really defining as herd immunity is from vaccination. So you create a certain number or proportion of individuals who are immunized and protected against a particular disease. So like Dr. Phil said, no, you do not want natural. Well, if it's a mild infection, then maybe it's good. But you know that COVID can kill, while only maybe more of the adults, but still it's an entirely different ballgame with COVID-19 versus the influenza vaccine. So let us not confuse natural herd immunity and vaccine herd immunity. We would like vaccine herd immunity if the vaccine becomes available, and we are really hoping that the vaccines that will become available will have high efficacy. Problema, I think the highest that we know we have read is about 60% in some of the vaccines, the preliminary data that we have. Okay, I think that I will end there.
Thank you, ma'am. We have actually another question for you. For participants who are enrolled into the phase 3 trial, what is the likelihood of developing unwanted side effects in the long term? What is the experience in vaccine development? And candidates may be biased to join because of the belief of definitely becoming immune to COVID.
There are many questions here. Okay, first one. Who are enrolled? What is the likelihood of developing unwanted side effects in the long term? So we will be following up, like with all vaccine trials, the participants will be followed up. At least for the Solidarity trial, we are following up to 18 months. Although I did say that the primary endpoints for Solidarity will be for 3 to 6 months, but all the other endpoints will be followed up to 18 months and maybe even longer. Other yung mga independent trials, so far as we have evaluated, they will run up to 1 to 2 years. So may parang long term naman. And then we have to consider that they will be required to do phase 4. And that phase 4 will be at least another 1 to 2 years. So maybe long term, but longer than that, I do not know. So maybe Dr. Philip can answer that. Kung meron pang longer than 2 years, 5 years. But anyway, sige, bago si Dr. Phil. What is the experience?
Thank you, ma'am.
Ano ba itong what
Thank you, Ma'am Nina.
sige,
I'll just answer your
sige, please.
sinabi niyo.
Please.
So some vaccines have ongoing surveillance, like meron 3 years, 5 years, even longer. So it's a state of continuous pharmacovigilance, and that's where regulatory authorities, monitor these events. So for adverse events, these need to be reported. So that's how it's being monitored for the long term.
Okay. So for most of the vaccines now, mga 5 years, pwede, no? So candidates may be biased to join because of the belief of definitely becoming, anong ibig sabihin nito? Becoming immune to COVID. Bias sila in the sense na it's positive for them, na magiging immune sila. Yung
Yeah.
Well, yes. Ano ba yung bias? Anyway, yung bias naman ay mawawala when we do randomization. Diba? Sino yung magpo-produce ng immunity, sino hindi, sino mapoprotect, sino hindi. So, I don't know about bias. Bias to join. We do not, everyone is voluntary ito lahat, no? Of course, everyone who joins will want to become immune, but that may not be the case. And we have messages already prepared that it will have to be explained to them that it is possible for them to get the placebo rather than the vaccine. So, and that is very clear. So, yun yung pinaghahandaan po namin. Because that's
just to add to that, in terms of bias, I think really what the participants in clinical trials will get is premier healthcare, because babantayan talaga sila ng qualified physicians, doctors, so they'll be really monitored closely. Their health will be checked, so they will get top-notch healthcare and being monitored throughout the clinical trial. So that's a distinct advantage. And clinical trials right now, usually we have active placebo. So instead of COVID-19 vaccine, may ibang vaccine na binibigay. So that's another pathway. But again, just to go back to safety surveillance, that's another phase of the study. It's called a phase 4 study. So that's more for the long-term active surveillance of the vaccine.
For COVID, I think the placebo will be another vaccine, although hindi naman din clear, it could be just the adjuvant itself. For some private companies, adjuvant pa kanilang placebo.
All right, we'll get to our next question. Our next question for either of our speakers is, how do you know if a vaccine has this binding effect similar to Dengvaxia? I think that's referring to Dr. Cruz's lecture a while ago. Is there a property to look out for, or is it just through observations in clinical trials?
So a mix of both, Nico. For the person who asked, you have to be in a state of continuous monitoring for these effects. And therefore, that's why you have the safety monitoring committee that you have monitoring the conduct of the study, and they meet regularly. And when there's a signal, even the faintest signal, and I've known that you've probably read it in the news that the study was paused. So it's a pause. It's not a stop or the study being abandoned. So it's really just a pause to look at the signals, we call it safety signals, whether they're related to a vaccine. And sometimes when they unblind the patient, turns out that the patient was not given the trial vaccine. So these are natural phenomena that occur. So it's also important to know what's the background rate, let's say, of a disease occurring in the country or the community. So it's important to note that, because most likely it's temporal association. Nabigyan ng bakuna, nagkaroon ng effect, so they associate it one and the other, which they couldn't find really a clinical connection to it. So yeah.
Yes, may I answer also? We look at certain parameters, like we prefer TH1 responses over TH2 responses. So when we evaluate, we look at those results from the vaccine candidates. And of course, yung sa observation in clinical trials, that's really part of the monitoring. Will the ones who get vaccinated, whether vaccine or placebo, develop the symptoms more severely or not? So we have those parameters in place. And like what Dr. Phil said, under the WHO Solidarity Trial, there is a global data safety monitoring committee. But locally, the WST also will be appointing 3 people to this local DSMC. That will be composed of one scientist, actually medical doctor, experienced in clinical trials, one epidemiologist, and one biostatistician. So aside from the specific IRBs monitoring the implementation of the trial, the local DSMC will also monitor, plus the monitoring committee from the FDA. So we have 3 bodies that will monitor the safety of the clinical trial here in the Philippines.
We have another question for Dr. Cruz. So congratulations, sir, for the excellent talk. What efficacy endpoints are being monitored, and how can those be obtained over such a short timeline, granted that the correlates of protection are not established yet?
Thank you very much. And the question came from Ma'am Ana Onglim. Hi, Ma'am Ana. Thank you very much for asking the question. So the primary efficacy outcome or endpoint we're looking at, and I'm basing it for the front runners of the clinical trials, is the first occurrence of COVID-19 via positive RT-PCR and symptoms, shortness of breath, and X-ray pneumonia. So that's the primary outcome. That's what efficacy is based upon. If you prevent that, that is an efficacious vaccine. And also, given a short time, we don't know yet the correlate of protection. That's why we have to look at all of these studies and submit it to regulatory authorities where they will map, because even the assays to measure this are different across all the trials. So it's really going to be a more kind of a regulatory challenge to determine these thresholds. But just to answer your primary efficacy outcome, it's prevention of disease.
Okay, we have another question. We have had a few colleagues who became infected with COVID-19, diagnosed by RT-PCR, yet failed to develop antibodies, hence were not made immune to the virus and therefore remain at risk. How then will the manufactured vaccine overcome this possibility?
May
Can I have a
I
go?
first answer that? This first statement here, colleagues who became infected with COVID-19 diagnosed symptomatic or asymptomatic or just an RT-PCR test. And then when you test them 2 weeks, 4 weeks, 2 months later, they have no antibodies. We have to look at the clinical history, the clinical data from these patients, because the RT-PCR tests are not foolproof. Some of them may give you false positives. So if they are asymptomatic, you know the CT value. Different brands have different CT values. So if a particular brand says 40 and below that it's supposed to be positive. If it's 0.1 or 1, then it becomes positive. Then you have to correlate everything with the clinical signs and symptoms before you can actually say it's true, or you can repeat the test. Normally, from an immunologic perspective, I would say that if there is true infection, there should be antibody formation. So if there are no antibodies developed, then I don't think that it actually got into that person. You know, the infection did not happen. So you cannot just say they did not become immune and therefore remain at risk. You have to consider the test use, the clinical signs and symptoms, you know, the time of collection of the specimen and so on. So it's not just one test based on one test, but a series of tests. So how then will the manufactured vaccine overcome this possibility? What does that mean?
I think I can answer that, ma'am, in looking at what the target efficacy rates are. So we're looking at the trials where they target 60% or even 50% efficacy and a minimum, let's say, of 30%. So even we have these numbers, when you translate it population-wide, you have thousands. That still is very significant. And also just to highlight that no vaccine or no therapeutic modality is 100% efficacious. So, you know, it's more or less kind of based on these clinical trials that we can determine the efficacy value and just project that into when it's being launched in the population. So you still remain at risk. However, it might be that instead of dying, you get hospitalized, but you live. Or instead of being hospitalized, you get a bad cold. So it's kind of like based on those parameters that we need to look. And that's why eventually the strategy here is not just going to be one vaccine. It could be two vaccines or one vaccine given yearly, could be like a flu. So there are these possibilities that we're looking at in the long run.
Okay, I think we have 2 more questions left, Brian. Oh, there. Can we post that question?
Again, yeah. Or we can go on ahead with a question for Ma'am Nina. Yeah, what are the incentives of having Filipinos participate in trials here?
That's always asked. Of course, even with the non-COVID trials, we give incentives to patients, usually in the form of transportation allowance or, you know, loss of the opportunity allowance. And I think at this time, noong araw, noong time ko, 500 yun. But I think this time, mga nasa 1,000. But we do not want to actually say that so much publicly kasi we do not want inducement, no? For the motivation to participate, but for the science of it and for actually believing that they are actually contributing to science. But there will be incentives. Ang magiging problem natin dito na pinag-uusapan namin sa sub-TWG, we're trying to standardize the incentives from the Solidarity Vaccine Trial. So that is government funded. Ang problema namin, baka yung pharmaceuticals ay magbigay ng sobrang taas na incentives. So we do not know if that will happen kasi we've had so many experiences before. You know how pharmaceuticals actually fund this trial. So we hope there will be some standardization. Of course, the ethics review boards are already looking into that. So kasi whether independent ka, private, or solidarity, they will all pass through the same ethics boards anyway. So medyo napag-usapan na namin yun that there will be some standardization there.
All right, and we have a few more questions, but I'm going to close it out with one last question for either of our speakers. Realistically, when is the earliest time that a vaccine will be able to ensure that it will be safe for all to return to pre-COVID lifestyle?
Philip, ikaw muna.
Ako muna, ma'am. So, you know, that's always a question that's being asked that can be answered in many ways. In terms of when will we get back to normality or a sense of our normality or live with the new normal? So, like I said in my talk, there are many factors to consider, not just a vaccine. So, a vaccine is not the only solution. It has to have multiple solutions in public health, disease control. So it's difficult to have a crystal ball and say that in 2 years' time we'll be back to normal. People will be traveling, because we don't know what the normal means now. Could be normal na nakamask na tayo all the time, or so it's difficult to say. But what I can just project is, with regards to a vaccine, most likely one will come out by before the end of the year. Some countries will have vaccine implementation programs early next year, but these are just for specific high-risk populations and not for mass vaccination programs. So we have to take it at that pace. It's difficult because we cannot predict even the uptake, because we did a survey here in the UK. 1 out of 5 will not want a vaccine. So that, again, is another obstacle because of that kind of belief and the hesitancy that we see. So there are a multiple kind of consideration of factors here, not just the vaccine, public health measures, educational measures, local government measures, and even the healthcare measures as well. So yeah, I couldn't give a very clear answer to that. I'm sorry.
Me too. I cannot even project, no? But yung vaccines should be available mid-2021 siguro. Under the COVAX facility, we may be able to get for 3% of our population the vaccines that will be found to be effective. So, and so far, ang schedule niya ay mid-2021. But that's only for 3% of the population. We're talking about probably increasing that to 20%. But the rest, kasi government, ano yung magiging ano doon, no? But we can look at also the government, the private sector pitching in here, making available the vaccines in the private sector so that we can have more than 20%, maybe 50%, 60% of the population being immunized. And that will be phasing, kasi the vaccines will not be available in a big volume at once. So yung availability niya medyo mahirap pa. And like Dr. Philip said, I'm a public health person. Hindi pwedeng yan lang. Ang solusyon hindi pwedeng always isa. Hindi pwedeng isa. Even if you have antivirals, sabihin na natin remdesivir, ganyan mga will work kapag may sakit. What still remains are the basic principles of hygiene, sanitation, yan. Yung mga paulit-ulit na sinasabi ng DOH sa atin. Pero ang problema, ang ating population, medyo pasaway. Pag konting, bumababa na, labasan na. Di ba? Tapos, of course, we have to think of the economy. But it's a very difficult, parang delicate balancing act. But we rely on everyone's help para we can work through it together. Iba-ibang aspects. Thank you.
Thank you, ma'am. Thank you, sir. So with that, I believe that concludes our grand rounds for this afternoon. To end this session, let us hear from the chair for the Department of Ophthalmology and Visual Sciences, our beloved and very own Dr. Marisa Valbuena.
Good afternoon. All throughout our long quarantine, we have been told the pandemic will continue until the time when an effective and safe vaccine has been developed and distributed throughout the world. Much has been said about how our lives will return to normal when a vaccine becomes widely available. While hope and optimism are very much needed in these very dark and trying times, it should be tempered with realism. With budget and distribution concerns, it is likely that the strategy is target vaccination of frontliners and the vulnerable population. Many people will still be susceptible to infection, and we cannot return to our pre-pandemic life for a long, long time. But it has been amazing how fast COVID vaccine development has been. But we still cannot throw our face masks yet. So with that, I'd like to thank Dr. Philip Cruz and Dr. Nina Gloriani and all of you who had actively participated in our PGH Grand Rounds. Good afternoon to everyone.
Thank you very much, Dr. Valbuena.
Thank you.
I would like to remind everyone to please sign the attendance and evaluation form through the QR code or URL posted. Do note that responses to the evaluation form will be accepted just until 20 minutes after the end of the webinar. On behalf of the department, thank you so much to everyone who joined us, our speakers, guests, and attendees. To end this very productive session, let us sing UP Naming Mahal.
Ooh.